Hello, everyone, and welcome to Research Ride, a 30-minute journey from idea to implementation of research programs in the Department of Medicine at Emory University. I am your ride leader, Charlie Searles, and I'm a faculty member in the Division of Cardiology and the Associate Vice Chair for Research in the Department of Medicine. Hey, Sarah.
Hi, Charlie.
How's it going?
It's going well. How's your week going?
It's going pretty good so far. What did you do for your birthday?
Happy
birthday, by the way.
Thank you very much. Well, my parents are visiting from Virginia, so we spent a lot of time just hanging out, eating a lot of food.
Nice.
And, of course, celebrating the 4-3 victory for Manchester United against Liverpool in the FA Cup quarterfinals.
Well, that was a big win then.
Oh, my gosh. When I say
It was at Trafford, right?
Yes, it was. And when I say I was so excited, I was shaking and almost just hit the ground crying.
Yeah, we have a real Man U devotee here. So what are you working on?
Well, we're getting excited for the Department of Medicine Research Day. It's going to be the 30th of October this year. And
awesome.
Yeah, and abstract submissions will be opening up within the next month. That's always a huge time for our office and for, I think, all of our faculty and trainees and students across the department.
Yeah. Well, we can look forward to that.
We can.
We can also look forward to our guest today.
Oh, I'm very excited for our guest today. How fun is that?
Bringing in the Department of Medicine research team, the whole team.
The whole team in the
here
studio.
in the studio. And so if our listeners haven't already guessed, we will be touring with Dr. Vibha Lama, who is the Vice Chair for Research in the Department of Medicine and our leader.
She is our leader.
Yeah, I'm really looking forward to this discussion. I think we're going to focus more on her research rather than her role as the Vice Chair for Research, but we're focused more on her research program.
Mm-hmm. She's a very successful researcher. We're very glad that she has joined us here at Emory, and I know she's going to have some excellent advice for our early career investigators and junior faculty.
I agree. So let's get going.
Let's do it.
Today we are delighted to have our research office team together in the studio. That means that we're touring with Dr. Vibha Lama, our leader, who is the Vice Chair for Research in the Department of Medicine. Dr. Lama is a physician scientist who is a leading expert in lung transplant biology. At Emory, she is the Professor of Medicine in the Division of Pulmonary, Allergy, Critical Care, and Sleep Medicine. She is the Augustus McKelvey Chair of Lung Transplantation Medicine, and she's Director of the new Comprehensive Lung Disease Program. Last but not least, as I mentioned before, Dr. Lama is our Vice Chair for Research in the Department of Medicine. You've been getting a lot of welcomes. You've been here for about a year now. So I won't welcome you to Emory, but I want to welcome you to the Research Ride podcast.
Thank you, Charlie. I'm very excited to be here, and I have heard the previous podcasts, and they are really wonderful. Thank you.
It's our pleasure. And I wanted to start off by asking you to tell us a little bit about about your research.
Thank you, Charlie. I could go on about this question forever, but I'll try to keep it short. So my work is in the field of lungs, predominantly understanding lung regeneration, repair, and fibrosis. It started off from the fact that I was very interested in understanding pulmonary fibrosis, a disease which is rather sad in that it's a little bit understudied, but even in spite of recent work on it, we don't have too much treatment for it. However, as I went through my journey, I became more interested in understanding lung transplantation, and specifically a very common problem after lung transplantation called chronic rejection, or in big words, we call it chronic lung allograft dysfunction, or CLAD. Interestingly, CLAD's form of a fibrosis, and that's where my interest and my journey came in this field. I think if I want to tell you a little bit more about my research, I like to say that as a physician scientist, I've always kept the focus of my research on the disease, and hence chronic rejection post-lung transplant is the center of universe of my work. But my work is very translational in a way that we take a lot of patient samples, we look at our patient samples, we try to correlate it to what's happening to our patients. And from there is born the whole discovery part of it, and which we then move into our lab and try to understand things better. With the goal really is to take it back to our patients and be able to make a difference in this disease.
So you got interested in lung fibrosis and diseases associated with fibrosis of the lungs, but you ended up focusing on, or have focused more on, chronic lung rejection, allograft rejection.
Yes, very true. So it's interesting, the two are tied. Since we don't have too many treatments for pulmonary fibrosis or even other chronic lung diseases like cystic fibrosis, COPD, a lot of these patients, the only viable option we have left once they develop chronic disease is lung transplantation. Now, Charlie, we've made a lot of progress in the field of lung transplantation, and our 1 year outcomes post lung transplantation are excellent in the field of around 90%. However, 50% of these patients will develop chronic rejection within 5 years, and hence the average survival post lung transplantation still continues to be pretty low, in the range of around 6 years, and lowest than any other transplant out there. So in a way, it's an unfulfilled promise. And, as a physician scientist, we're always motivated in our work by our patients. So I have had patients who got transplanted at a young age of, say, 20s because of disease like cystic fibrosis. And unfortunately, by 25, 26 years of age, they have chronic rejection, which is fatal. So that's where my interest in chronic rejection came from.
That's very interesting. I wanted to ask how you became, or what was your path to becoming the McKelvey Chair of Lung Transplantation Medicine at Emory? Where did it all start, and how did you get there?
So it started off by a person who actually was not clear in what they want to do, and I think it will come to this question of how we think about research when we are clinicians or when we are physicians. My journey has been, I started medical school at age 17 because I went to medical school in India. And by age 21, I was actually in England pursuing higher education. And then when I came to the US and I did my residency and then my fellowship in pulmonary critical care medicine, I was a pretty nicely baked doctor, a physician, right? And I didn't have much research experience. However, I always wanted to be the one who thinks about problems and wanted to make a difference in some of these diseases where we don't know some of the answers. So when I went into research in my fellowship, I had the opportunity to do basic science or laboratory-based research. And I worked on these lipid molecules called prostaglandins, which are everywhere, very important in a lot of diseases. And loved it and hated it. It was interesting, but I also remember I'm a clinician. I want to keep going back to patient-oriented research. With that in mind, I ended up doing some training in clinical research as well and got a master's degree in statistics and clinical research design. And then decided to marry the two by saying I want to bring my patient-based samples back and understand them. And it was the emerging field of translational research, which is now extremely common, but was an emerging field of translational research. And from there became this program, which became, with the center being our patients, who it's been very fortunate in my research that our patients are so committed to also giving whatever they can to research. Remember, these are the patients who have had lung transplant. And I'm going to give you one example, which is the hardest, which is when I have sometimes patients who are in the end stage, and I go to them and I say, what can I do? And I remember this particular incident of a young mother who said to me, Dr. Lama, after I pass away, I want you to take my lungs and learn from my lungs. So that's exactly what we are trying to do, is that we want to honor our patients' wishes for them to also make a difference in the disease they had. And hence the whole translational part was born, in which we have patient cohort in the middle, samples coming in, and then understanding. And from there came the field of the lung stem cells. And we discovered a stem cell in our patient samples, which was not known about. And these are called the lung-resident mesenchymal stem cells. And over the last 20 years, we have understood a lot about these stem cells and how they participate in lung repair, fibrosis, how they can be used as biomarkers of diseases, and how we can make new drugs based on how these cells are participating in fibrosis. So sorry, a long-winded question to how I came about this field. And being a McKelvey Chair at Emory is a wonderful opportunity to continue to make a difference in the field of lung transplantation. We are very proud of our lung transplant program here, which is the only one in the state of Georgia. And we really want to further expand this program and be known as the national leaders in the field.
Well, congratulations. It seems you're well on your way to do that. From the research side, the way you're telling it now is that you were a clinician first, but you understood, or you developed this desire to become more investigative as a clinician, and that's how that's what led you into the research lab.
That's absolutely right, Charlie. I think we are very fortunate that as physicians, when we see our patients, and I know that as our cardiologist, there are many times we are sitting in the clinic and a question comes up in our mind, and we say, why am I seeing this particular, pattern of disease? And to have that ability to go and investigate that, whether it's through your clinical cohort you have, or clinical database you have, or being able to take some of the patient samples back and be able to investigate them at the cellular basis, or to be able to study them in models of the disease, we're extremely lucky in that. So I feel very fortunate in us being able to merge the two sides of being able to ask a question and then be able to answer it as well.
So this brings in similar themes to other physician scientists that we've talked to, right, Sarah? I mean, our last podcast, we had a discussion with Dr. Sands, who has a very similar, I thought, description of how his research career evolved from a clinician to an investigator. Let's go back to lung transplant. And you said that lung transplant might be described as an unfulfilled promise. What do you mean by that?
So, Charlie, lung transplant is done to extend life. We always say that we don't just do it for quality of life, we do it to extend life. So when we choose patients for lung transplantation, we look at what would be the survival without lung transplant and how lung transplant will prolong their life. However, unfortunately, because of our median survival, so 5-year survival of around 50 to 60%, we are giving them only a finite number of years. That being said, I must say that I have lung transplant patients, my own patients, who were now 10 years out, 15 years out post-lung transplant, right? But that's a minority. We have around 20% of the patients who will go beyond that time. So it's an unfulfilled promise, because we would like to have not just put them through this procedure, which is difficult. It's a lot of emotional and physical part which goes into it. But then to be able to give them a very long, wonderful life to live after that. And the major barrier in that is chronic rejection. Hence, a lot of our focus is understanding this problem of chronic rejection and understanding how we can prevent it and treat it.
It must be, it takes a lot of effort to figure out who is actually a good candidate for lung transplant, given the length of the median survival for lung transplants. Why is that, though, that the lung transplant is so much different than other solid organs like the heart or the kidney?
That's an excellent question, Charlie, because like I mentioned before, our survival post-lung transplant is unfortunately the worst among all solid organ transplantations. There are many factors that go into it. The first factor is that the lung is a rather complex organ. And I'm not trying to make fun of heart here, Charlie, as you as a cardiologist, but the lung has more than 40 different cell types in it. It's a very, I call it an amazing structure because of the carbon dioxide oxygen exchange units. But more importantly, remember the lung is constantly exposed to the outside environment. We heard a lot about it at the time of COVID when lungs were the ones which had the major brunt of the disease, right? So as an organ which is constantly exposed to environmental factors, infectious insults, the immune system reactions can be much stronger than other organs. And when we are transplanting a patient with a lung which is a different allotype, what we call is there is an immune mismatch, right? Now, this lung being already primed because of that, now getting exposed to outside environment, there can be much worsening of rejection and things like reflux disease. We talk about reflux disease in which there's acid reflux, which people are very aware of. It's considered one of the factors which contribute to chronic lung allograft dysfunction or chronic rejection. Viral infections such as cytomegalovirus infection, those can also exacerbate or lead to more rejection post-lung transplantation. So because of these complexities, the second thing I like to say is that as an organ, we have to understand more about all organs, but in the lung, a lot of our work has been focusing on understanding the resident stem cells. And, there are a lot of, a major premise of our work or major contribution of our work was bringing the understanding back to the fact that the lung has its own resident stem cells, which are there from the time of embryogenesis or at the time from the fetal growth of the lung, and that these stem cells are the ones which are very important in local regeneration, repair, and also disorganized repair, which is fibrosis. So we think about these local communities in the lung much more because of some of our work as well, in which we describe this stem cell called lung resident stem cell and how it interacts with the surrounding cells. Think of it as a community of mesenchymal cells, epithelial cells, and that these mesenchymal cells then also signal the immune cells, which are the ones which are causing the rejection. So I think a lot more work needs to be done in understanding these local communities and how we can change these cells or impact them to improve repair and less disorganized repair or fibrosis.
That's fascinating. The resident stem cells, these mesenchymal cells, are in the donor organ, the organ that goes into the transplant recipient, and they are being activated to help manage the immune adaptation of the new organ. Is that
absolutely
accurate?
Beautifully put, Charlie. Yes.
Is there a way to predict from the donor whether their mesenchymal stem cells are going to overact or have some of the detrimental effects that I think that you're alluding to, that you need some of the mesenchymal stem cells to do their thing to help with repair of the new organ as it adapts to its recipient person. But I think you're saying that if these stem cells can be overactive and they can promote fibrosis.
Very true, Charlie. So it was a very interesting discovery. So I'll tell you a little bit about it because I think it'll be fascinating for our audience and especially for our younger faculty or fellows. So this started off as us looking at lung washing. So we do a procedure called bronchoalveolar lavage, in which we go in through a scope and put some saline in and wash out, and we get cells back from that. In our lab, what we discovered was that when we are taking some of this extra lavage back to the lab and letting it sit there, we start growing these cells out called mesenchymal cells, which we were not aware that they can grow from lavage before. As we understood these cells better, we understood that they're kind of a multipotent stem cell, so they can give rise to different cells. But what was fascinating was that we could now look at male patients who had a female lung or female patients who had a male lung, because when we transplant, we don't match by gender or sex. So we could look at these cells and say, 15 years post-transplant, are these cells donor or recipient? And when we did that experiment, we were shocked that they were still donor. So the donor cells were still very much alive and very active in these lungs. Then we started looking at the number of these cells, and we realized that these cells go up in number early post-transplant when they are helping with repair and regeneration, and then they go back down. But if they go back up later on, post-lung transplant, then they predict development of chronic rejection and are participating in the process of chronic rejection by exactly what you said, by being overactive, by affecting the immune cells, but by also causing scarring. Remember, these are the cells of scarring. They make collagen or other sort of extracellular matrix material, which stiffens up these lungs. So yes, there are a lot of things which we can do. There are, factors which can help us understand when this cell goes from what I call a good cell to a bad cell, right? What are the changes which have happened and how we can reverse those changes? Are there predictors before transplant? Like, could there be some genetic factors which makes these cells more immune to developing into more of a fibrotic cell? So a lot has been done, and a lot more we are planning to do, which we hope will help us understand not just chronic rejection, but in general lung repair processes as well.
I just want to go back 1 second here. We have been talking about CLAD in general, chronic lung allograft dysfunction.
Yes.
And there are subphenotypes of CLAD, which I wanted you to talk about a little bit.
Thank you.
But
yes.
I wanted to also ask about, there are other types of rejection, right? As far as primary rejection, acute rejection, and then you have the chronic rejection.
Yes, very much so. So, post-lung transplantation, there are multiple different complications which can happen. The very first one is primary graft dysfunction, which is right after we put the lung in. There might be some injury which has been caused by what we call as ischemia or a reperfusion. Because remember, we're taking the lung out, pretty much putting it in a bucket of preservation solution and bringing it in, right? And sewing it back in. But I say lung's an amazing organ because it regenerates beautifully. And but the first complication is primary graft dysfunction. Second is acute rejection. Acute rejection is diagnosed mainly by biopsies or decline in lung function. And then the third complication is chronic rejection. Now remember, chronic rejection is linked to both primary graft dysfunction or PGD and acute rejection because both of them will predispose the lung to developing chronic rejection. In chronic rejection, Charlie, you asked about the two phenotypes, and that's actually a very interesting and important question because we used to lump everything together when I started research in this around 20 years ago. And over this time, we have understood that rejection can have two different phenotypes called bronchiolitis obliterans syndrome and restrictive allograft syndrome. But the most important thing about them is that they are present a little bit differently. The radiographic features are different on high-resolution CT scan, and their progress is different. Patients with RAS have a much poorer prognosis than patients with BOS, but within BOS, you can have some patients who do worse than others. And, one of the works which we have done recently is that we have developed two different models of this disease. And then when we compared these two models, what we realized was that these two phenotypes have very distinct pathogenesis. So one is more related to cellular immunity and the other to humoral or antibody-mediated immunity. Now, that's very important because that could explain why some of the drugs which we give these patients don't work, right? Because if there are two different types of phenotypes or diseases which we have lumped together, then we need to be careful in our clinical trials or when we are thinking of targeting these diseases. And that's exactly the importance of all the wonderful translational work which is being done in these areas and clinical work which is being done in these areas. And as time goes on, I think we are thinking of more and more what we call as a personalized approach or a more approach based on the specific patient rather than, one size fit all approach.
So it is my understanding that BOS, which is bronchiolitis obliterans syndrome, has a better outlook than RAS, which is restrictive allograft syndrome.
That's right. So 70% of the patients with chronic rejection will fall under the BOS umbrella and 30% will fall under the RAS umbrella. And
we don't have much to offer to the RAS patients.
We don't, in a way that both these diseases, unfortunately, at this time, we don't have much to offer them. But patients with RAS, the median survival is very low, less than a year. So that disease progresses very quickly, right in front of our eyes, and it's very distressing as a physician actually to see that. And so a lot of work is being done in understanding RAS. As I said, some of the work from our laboratory has shown that this is more related to humoral immune responses by very clearly showing that if we take away the B cells or the antibody production, we do not develop RAS-like pathology. And in patients too, we know that patients who are developing antibody-mediated rejection are more prone to get RAS. So now we can think about specifically targeting antibody-mediated rejection or molecules which are associated with it in patients with RAS with the hope of stopping the progression or even preventing the development of RAS.
Very fascinating the way that you are able to model human disease in your animal models and potentially identify new therapies and ways to manage chronic lung allograft dysfunction. I wanted to go back quickly to the question of your mentors, or who most influenced you in your path here towards the McKelvey Chair of Lung Transplantation.
So, Charlie, one of the advice I give junior faculty, and I know you have a question on that because I have heard you ask other presenters that question. But one of the things which I tell my junior faculty is that you should seek your mentors. And that's what I've done throughout my career. I've sought mentors, and I think at any stage in your life you need mentorship. I started off with excellent mentors on the side of basic science and clinical research, but I didn't seek just one mentor. I sought multiple mentors. And then I had mentors all along my career who have guided me in thinking about different areas as I have moved through my career, right? Like I started off by understanding basic science, clinical research, marrying it into translational research, understanding how to develop a program, how to set up a program around our lung transplant patients, then going into more mechanistic animal modeling work. And all throughout my career, I have had a fascinating range of amazing mentors. And I don't even say I can even put all the credit on one mentor, but I would say I have been mentored by, they say it's a very common quote that you have a village to raise a child. It's exactly the same. I have had an amazing series of mentors at Michigan and beyond Michigan as well, who have guided me.
It seems with the direction or the approach of your research program, that is absolutely required that you have multiple mentors or people who can advise and guide in different areas of your research program, as particularly as diverse as your research program, involving clinical samples, animal models, clinical data. That would be pretty much essential.
And Charlie, I say along with the mentors, there are the collaborators, right?
Yeah.
And I believe a lot in, I say the word convergent science a lot, but we want to bring novel technologies into our work because as physician scientists, what's our goal? Our goal is to impact our disease we are studying and to improve the life of our patients. So I have always reached out and gone to people, made phone calls, picked up the phone, gone and stood outside their office. If I needed their expertise or their help, I go get it because I do it because I'm doing it for my disease I'm working on. But I think I would also like to say here that, my second part, which I said, seek your mentors. But the second part is, don't be afraid to divert from the path. When I started in this field, translational research was a completely new path. I wasn't supposed to do that. I got into a little bit of trouble wanting to do both basic and clinical research. But, there's a Robert Frost poem, The Road Less Taken, The Road Not Taken, I think. It's about the path.
I think it's less taken.
Less taken, the path which is not,
Yeah.
And
Yeah.
I think something's very beautiful about that poem. And I always tell junior faculty, don't be afraid. Go after your passion and then go and carve a path for yourself.
Okay, well, you stole a little bit of our thunder because we're coming now to the portion of our program that we are calling now Do or Don't. So the question that we wanted to ask you, and you've partially answered, is what is one thing that you did early in your career that you would advise others to do? And then the don't is, what is one thing you did early in your career that you would advise young investigators not to do, or early career investigators?
So I will just read the last paragraph of this poem for you. I shall be telling this with a sigh. Somewhere ages and ages hence, two roads diverged in a wood, and I took the one less traveled by, and that has made all the difference. So this will be my advice for our.
That's Robert Frost.
That's Robert Frost, yes. And your question of what not to do, right? And I think the biggest lesson for that is not to let your focus be taken off, because I think early on in the career, having a more focused approach is extremely important. And there are many times when we are pulled in many directions, and I don't know about you, Charlie, but I want to always do a million things at a time, and I get excited about multiple ideas. But early on in your career, it's very important to keep the focus and get help in keeping you focused. I remember I had a wonderful division chief at Michigan, Galen Taves, who was our pulmonary division chief. And Galen was like a parent. I would get a scolding all the time if I wanted to get outside and do more. And I would get very upset at that time with him. But many years back, I look back and I say, thank you, Galen. I'm glad you said it. So I think listening to your mentor or whosoever is the person who is going to guide you in your career is very important.
Thank you for that advice. It's well taken. Another question that we ask is about what kind of activities do you enjoy outside of the lab or the clinic?
I don't have very exciting activities. I'm going to be the most boring person in answering that question. However, I spend a lot of time with my little dog. He's a Bichon, and his name's Omi, and I think this is my first time having a dog. He's been in my life now for 6 years, and I think he's made a tremendous difference because he gives me that calmness which I need at the end of the day, going out for walks with him. I love gardening. Again, I think it's very helpful because I'm not a very patient person.
Flowers, herbs. What is your
vegetable gardening?
Vegetable
I love
garden.
vegetable gardening.
Vegetable. What kind of vegetables?
So I grow everything from mustard greens to spinach to my tomatoes never do well, Charlie. I don't know why, but other than that,
They should be doing well in Georgia.
exactly. And I think one of the excitements about moving to Michigan to Atlanta or Ann Arbor to Atlanta is the fact that I'm going to have a much bigger gardening season. So I'm very excited about that. And I'm a foodie. I love going to different restaurants. And again, Atlanta is a great place for that. So I think I'm advertising Emory and Atlanta by saying you will have more time to do gardening and you will have more restaurants to have wonderful.
And, it would be farm-to-table restaurants, right, that you can enjoy here in Atlanta.
No, I've been very impressed by the food scene in Atlanta.
Okay, it's now for our last segment, which is Research Ride Playlist. So when you ride your bike, sometimes you might put the earbuds in and listen to some music while you're riding. And so we've been developing this playlist and asking guests to chip in suggestions as for what they would want on this playlist.
So Charlie, I'm a little tone deaf, but I do love music, and I do listen to a lot of Hindi and
nice.
Punjabi music as well. So really, I could give you some great recommendations for that part as well. But at the same time, I think there are two songs I would like to add to your playlist.
Okay.
They're both going to have very important meaning. One is Location by CLAD. I love that song. There are two words in it which keep coming up, which is location, and I think that's about...
the artist is Khalid?
Yeah, Khalid. Oh, you haven't heard Location by Khalid?
No. Have you heard that? You have?
Yeah, of course.
shoot. Okay.
So there are two things in that song, the location and the communication.
We're going to cut that one out, I guess.
Oh, no, we're keeping that. We're definitely keeping that.
And those are very important in research.
Okay.
Charlie, location.
Yeah, sure.
Where you end up making a career for yourself, and communications.
Yeah.
Right? And the second is Favorite My Husband. And he played it a lot when we were dating. But at the same time, I think it's very important in research because we need to have faith when we are doing something. And it can be sometimes a very long road in research. And
Favor My Husband.
It's a favorite song of my husband as well.
Oh, favorite. We're building up.
Yeah, so
we're
the song is...
We'll cut that.
The song is Faith by George Michael.
Oh, okay. I know that one.
Yes, I knew you would know that one. Sarah,
Gotta
do you know
have
that
some
one?
faith.
I think I'd recognize it if you played it, but I can't hear it in my head at the moment.
Oh.
Wait, so you don't... George Michael
I
is
know who George
too
Michael is.
too old for you,
No,
I
no,
guess,
no,
right?
I know who George Michael
So
is.
good. So I picked two songs, one which Sarah knew and the one which you knew, Charlie. So that's
okay.
good.
All right, good. Yeah, perfect. Well, Dr. Lama, Vibha, thank you so much for joining us today on this research ride. It was a fantastic tour of your research program and some of the controversies or some of the issues with lung transplantation. And I want to thank our listeners for joining us on this research ride. This has been Charlie Searles. I'm your research ride leader. The producers of this podcast are Sarah McClellan, who's also our studio engineer, and Ben Searles, who's also the podcast editor. The theme music has been composed by Hannah Searles. And thank you very much, and we'll see you next time.